So Birol, last but not least, I'm gonna introduce Birol Aydin. He is a clinical senior embryologist and scientific director at Ovogene Bank with over 15 years of experience in embryology and embryology across 20 countries. He has set up 10 IVF labs and specializes in donor egg banking and oocyte cryopreservation. He has conducted over 50,000 ICSI embryo transfer cycles, 20,000 PGT cycles, and 15,000 egg donation cycles, as well as 122 spindle and nuclear transfer cycles, achieving 22 pregnancies and 12 live births. Birol has published more than 15 scientific papers and contributed to advancements in genetics, certified oocytes, AI for oocyte morphology, and time-lapse vitrification imaging. He is also an editor for Elsevier cryo guideline. So today, Birol will share Ovogene's experience with the ultrafast vitrification and ultrafast warming protocol, showing us not only survival rates, but also clinical outcomes and the metabolic and transcriptomic analysis. So please, Birol, we are ready when you are.
So thanks for kind introduction. It's a pleasure to be here. So we already listened to a great presentation, which shows on the backstage how ultra-fast, ultra-rapid vitrification and warming working from the genetic side and also morphokinetic side. So I will go a little bit from different perspective. So we will go for transcriptomic, metabolic, and also for each stage of embryo development for the morphokinetic stage. So as we know that vitrification avoid intracellular ice crystal formation, which create that and maintain that viability. And also we are trying to keep cryoprotectant concentration as safe as possible for the toxicity perspective. So the last 10 years show us that vitrification protocol, we are trying to optimize with a different way, and we are trying to improve efficiency either for oocyte vitrification and warming, also for the embryo, while especially for egg banking get a very critical level, so many egg donation cycles, and we are using a cryo egg mostly, and for us survival and embryo development is very critical. As we start for every new techniques, lab technologies, we should always ask the question if those techniques or technologies are for patient benefits, is it safe as clinically, or we can improve this as much as on the scientific background. So I always like this diagrams, which shared by Joyce Harper. So when we accept some hypothesis for new technologies or techniques, the first thing, of course, we should create a study. And after this study, so in the animal model and then human model, and then different the genetic metabolic perspective, that finally when we believe we have already evidence that's safe, then we can start to use and take for the clinical practice. So we know that already there are questions about vitrification, especially for frozen embryo transfer. A Chinese group published that they have concerns that for new genetic mutation, which is connected with the cancer, which was scary. Of course, that we have to ask this question as a scientific perspective. So ultra-rapid vitrification and warming, so we can avoid the risks for these new technologies as a low survival rates, the risk of ice crystal formation, especially from the toxicity side, osmotic stress risk, and of course, we need innovation to optimize these technologies. So how ultra-rapid vitrification working? As we have a quick cooling rates, we have mechanism of action which we can improve from the theoretical and practical part, especially for limited experience of the embryologist can get benefit from these new technologies. And of course, why we are reducing oxidative stress, also we are reducing risk of toxicity with ultra rapid vitrification and warming. As we start years ago with the slow freezing, and then we were using ice crystal formation, and then with vitrification we start to use a glass-like state, and with ultra fast we pass immediate glass-like state, which increase the efficiency. As we look the description for between standard and ultra fast vitrification, there are no much difference actually for the composition of the media which we are using as equilibration and vitrification. There is time difference, how we became to this position to use the same concentration of media but different timing, just the way that we find out between exchange between cryoprotectant and the water. So it's limited with in equilibration with the 60 seconds. So when we find out this question, this answer for our question, so that was easier to use ultra rapid vitrification, especially for oocyte freezing or collapse embryo freezing, zygote freezing, even day 2, day 3, and day 4 stage embryo freezing to keep equilibration 1 minute. It will give even same or higher efficiency as a survival comparing regular conventional vitrification technique. The same thing for ultra fast warming. Once we have 1.0 or 1.2 molar trehalose or sucrose concentration in 37 for 1 minute. So we find out for again collapse embryo, oocyte, zygote, day 2 and day 3 stage, all embryos can give higher survival rate only using of the thawing, basic thawing solution in 1 minute. So as typically when we start for embryo, you will see on the video during the warming and the embryo stage and the oocyte stage. So if you take this, record this video with the regular conventional warming and ultra rapid warming, you will not find out a difference, big difference in the timing of 15 minutes for vitrification, 15 minutes and 1 minute, and for warming 1 minute, either if you use the DS solution and washing solution 3 minutes plus 5 minutes, you will get the similar video record from oocyte forming. As you see, for us, most important, which we are keeping cryoprotectant mostly around the perivitellin space between cytoplasm and the zona pellucida. When we exchange this, so that means that we are saving the oocyte as expanded version for next step of oocyte or embryo culturing. So of course, studies are important while this technique is very new and we are trying to optimize and develop. So the different publications shows that with these techniques, we can reduce osmotic stress. Also, this did not disturb a meiotic spindle as we have the similar result I will share and to let high blastocyst rates. So as I said, especially for warming, one-step oocyte and blastocyst forming, we are using 1.0 or 1.2 molar trehalose or sucrose, which we are using with 300 microliter exactly to keep temperature optimization. So you will see here with different type of oocyte from a patient from different donors as which we vitrify with ultra rapid and we thaw back with ultra rapid for 1 minute. So as different publication focused on mostly clinical. So the first publication shows ultra rapid vitrification for 1 hour post-forming. You will see for mature oocyte between conventional and ultra rapid. As always, ultra rapid give higher survival rate. The same things you will see for the spindle configuration and location. So after ultra rapid forming, they find out the spindle configuration didn't change and spindle doesn't have any morphological damage as we got the same result in our study. So the different study worked with the GV germinal vesicle model. So the compare with the fresh conventional ultra rapid vitrification. As for the GV version, M1 and M2 version also 24-hour and post 48-hour of the warming and freezing, they find out ultra rapid give better survival rate. As also studies for the clinical practice, they show comparing regular vitrification and warming significantly higher clinical pregnancy rate, implantation rate, and ongoing pregnancy rate with ultra-rapid vitrification and warming. So if we check our studies, which we are talking about multi-center studies, while we are egg bank, we are sending many oocytes, frozen oocyte, donor oocyte all around the world, and which we get from different embryologists, different experience of the embryologist, different lab environment, and different culture media. So they used approximately 22 clinics, IVF clinics. They use ultra rapid warming, and with the conventional, with the sibling oocyte, we get 94.5 survival rate, as fertilization and cleavage is optimal. As you see, 39.6 blastocyst rate and 61.4 euploid blastocyst rate over 55 clinical pregnancy rate. When we back to ultra rapid vitrification and the clinics warm by ultra rapid, so we already find out very high survival rate. The fertilization rate is a similar, but when we get the blastocyst rate is higher blastocyst rate comparing conventional. While this is not because of the, again, connected directly to technique, but while we are increasing cumulative survival rate, of course, we are increasing blastocyst rate and euploid blastocyst rate as well. While we have morphokinetically higher quality of the blastocyst rate, as you see, clinical pregnancy rate also is higher. So if we are searching some limitation, of course, still there are some limitation which we are looking on it. So firstly, the expanded blastocyst. So still there are concern if technique is totally available for expanded blastocyst, while blastocell containing big structure and more liquid comparing oocyte. So that's why there is a question for expanded embryos. So some studies and some researchers still supporting that for expanded blastocyst also available. So I still have concern for that. But generally another limitation is there are limited number of study yet. So we need more study and more research. So how we were working for metabolic and transcriptomic analyzing. So we worked that non-invasive evaluation for the metabolism and secretome for human embryos, which is undergoing ultra-rapid vitrification. We used infrared spectroscopy and RNA sequencing to identify biomarkers for healthy embryos. So we already collect 1,214 donor oocytes, the sibling oocyte, half of them with the ultra-rapid, half of them with the standard protocol we freeze. Then we tow back with the half of them with ultra-rapid and half of them with standard conventional warming. Then we fertilize them and we create embryo. So after that, we were checking secretome profiling, so lipid profiles, nucleic acid profiles, and the protein profiles. They spent the time in culture till day 6. At the same time, as a clinically, we collect oocyte survival rate, fertilization rate, day 3, blastocyst rate, and euploid rate. And the final stage was RNA library. So for spectroscopy, we use the old samples, which we got mostly from the culture media, which embryo developed on the different stage, on the stage day 0, day 1, day 3, and day 5, and we analyze their metabolic levels. As a metabolic result for different stage of the embryos, we find out the similar metabolic development, as I will show. So between fresh, ultra-rapid, and conventional vitrification and warming. So material shows the similar DNA concentration, similar lipid concentration, also protein structure, nucleic acid structure, and the protein concentration. So this is a good point for us that understand the fresh cycle, fresh material, conventional vitrification and warming material, and also ultra rapid vitrification and warming material has similar outcome. So as you see for the zygote, also they have similar diagram for day 3 embryos as well. As if we come for the transcriptomic data, we use the fresh, ultra rapid, and conventional. We got biopsy and we used the whole oocyte for transcriptomic analysis. So this is how we create the library for gene expression. So actually that we have to understand here that to create a transcriptomic data or RNA library, it's quite difficult. Even if you focus on the cell biology, you will find a very limited number of study which exactly achieve that to create a RNA library. So it was really difficult to create this. So according to this, we compare ICSI time for the conventional and ultra rapid. So how oocyte give reaction after ICSI. So because this is also concerned that most of the oocytes still get degeneration after ICSI even if they survive. So we got analysis for different thawing methods for transcriptomic perspective. As you see here, the post-ICSI degeneration, we were comparing with the ultra-rapid and conventional. So ultra-rapid show very standard diagram, but conventional show so much up and down. So that means doesn't matter the quality of the oocytes or if they survive. So conventional is giving much higher degeneration after the ICSI, post-ICSI. So the same things if we check day 5 blastulation rate. So ultra-rapid shows very good quality of the morphokinetic diagram comparing conventional. Same thing for the high quality day 5 blastulation rate and day 6 blastulation rate as well. So if we check the clinical pregnancy and the live birth rates between conventional and ultra-rapid, so we will see that ultra-rapid have significant higher result comparing conventional. If we come that more morphokinetic diagram, so we will see the survival rate almost a 10% difference because when you compare 1,400 oocyte and create for them the embryos, so data is more accurate. And I have to tell this is a multi-center study, so it's not from the one lab, just from many different lab and many different embryologist who has different experience. So which is the important here, post ICSI degeneration, we find out for the conventional 5.7, like comparing ultra rapid very high. So also high quality day 5 blastulation, it's a very high difference and significant difference between ultra rapid and conventional. So what is the interesting? So we find out day 6 blastulation higher in the conventional comparing ultra rapid. So here have morphokinetic changes. So we find out for ultra rapid, we have a more early cleavage 27 hours, which is a good point for embryo selection for implantation. So comparing conventional, ultra rapid is giving much better morphokinetic development, especially that if we compare the embryo quality on the morphological analysis. So the AA and AB quality embryos almost significantly higher in ultra-rapid group comparing conventional group. As transcriptomic, we were using that heat map technology as well. And we had a number of reads ranging between 20 to 50 million. So we already get 98% of the sample. We had reading, successful reading, which is very high. So in the first diagram, you see the down, the oocytes, as you see, fresh oocytes with a different color, which is showing the blue color. With the orange color, you can see ultra rapid, and the brown, you can see the conventional. Those are from biopsy material, and they are in the same place, in same location. That's the similarity test. So that means they have similar matrix of gene expression value. So the same thing on left side on up for all sides with the purple, green, and red color. So they are showing fresh, ultra rapid, and conventional oocyte freezing and warming. So you can see that they have also similar gene expression. So here that pairwise similarities in the dark color, which you are seeing, showing the similarities of the groups. So here as well, we get same transcriptomic profile as a gene expression. So that shows that both three techniques, all three techniques have similar gene expression level. As a conclusion to ultra-rapid vitrification and warming methodologies, which allow faster cell dehydratation and rehydratation, so which is the potentially giving higher survival rate and the clinical outcomes comparing standard and conventional protocol. So for shortening the time is giving chance to us to maintain lab work. Also, it brings that if we have a limited experience in the lab, so to keep optimization for the survival and embryo development with the cryo oocytes especially. One of the important things, we believe that we are keeping much safe the toxicity. But here, one thing you need to know as a clinical practice, I'm saying that. So after warming, especially oocyte by ultra rapid with warming technology, so we always suggest to use additional 10% of human serum albumin in culture media to keep these oocytes after warming till you make ICSI. So why we are doing that? Because we know that protein structure is a very good to absorbing toxicity. So especially after warming with ultra rapid, because we have very short exchange between cryoprotectant and water. So high percentage of albumin will be very helpful to keep zona permeability and also to give you higher survival rate post-ICSI. And also the one-step rehydratation protocol allowing that the result which you get higher survival rate will give as a cumulatively higher blastocyst rate and the clinical pregnancy rate. So here the most important point that we have to understand as a two-way we have for the one new techniques or protocols to accept it as a safe for our patient. The first one, we can wait a long-term clinical outcomes, which is pretty risky, of course. So we don't know if those babies which will come from this technique will have any kind of risk, life risk, gene mutation. Of course, this is very critical, or we will go to analyze transcriptomic or metabolic way on embryonic stage. So that's why we were trying that effort, while to go directly with the clinical way and to use for the patient embryos and material immediately. So it was pretty risky for us. That's why we start from this stage. And then, of course, now we are insured, and I'm sharing this data with all of you, and you can be insured this ultra-rapid vitrification and warming is showing safe metabolic and transcriptomic profile for biological material. So we already developing this technology under European Union support and foundation. So we are trying to create the big study group for transcriptomic profile to check morphokinetics on the zona pellucida and also the spindle stage, and morphokinetics embryo development will be safe. And the final stage will be the new device validation, which will optimize, because now the device validation will be very easy, while we will use only single step of warming and vitrification. So thank you very much for your time and attention.
Thank you very much, Birol. So amazing presentation, very complete, very informative. Thank you very much.